Yes. Yes.
Okay, great. Great. All right. Sorry about that. We can get it started. Any questions about the material from last week, or so we're doing the exam next week. And this is going to be, as we talked about it, this is going to be in the class. All of you will show up to the class. We'll print the exams and then we'll bring them there, and then you'll write them there, pen and paper, so you can kind of be ready to write. They're going to be a mix of multiple choice questions and essay questions. I will share a study guide with you all after the class. This is for the exam. There are going to be around 10 short essay questions on it. You're going to study those, and then you're going to get a subset of those as the short essay questions on the exam. But there will also be multiple choice questions from the other material that we covered in the class that are not necessarily mentioned in the study guide. So the study guide is to make it a lot easier for you to study for the short essays and general understand the material, and then the multiple choice are really for you to make sure that you understand all the material. So that's how the format is, and I think it works well. Any questions about that? No, all clear?
One question.
Sure.
What percentage of the whole exam will be the multiple choice versus the essay questions? What ratio type of thing?
Say maybe 40-60, maybe we have 40% of the grade will be from multiple choice and maybe 60% from short essay, give or take.
Okay, thank you.
Yeah.
Yeah,
go ahead.
My additional question was, you said that the study guide is going to be for the short essay questions, not for the multiple choice.
Yeah.
Got it. Thank you.
The essay questions are more basic. They are supposed to increase your basic understanding of the material. The study guide is basically asking you questions to think about different things. They also cover a lot of things that you will need to answer the multiple choice questions. But it's not the whole thing. I want you to actually study the material outside. I don't want this to turn into you memorizing some answers from the study guide and then coming to the exam. I want you to actually put the effort to understand what they're trying to teach you. Yes, Zoe?
I was just wondering how long the short responses were going to be.
The essay questions?
Yeah.
I think as you get the study guide, you'll see what they look like. They're usually maybe half a page or a little more than that. It really depends on how efficient you are in answering questions. But the goal is that you'll write as much as it takes to answer the question without being too verbose or without being too brief, right? You just have to hit the right spot. And I mean, the TAs will walk you through. So the idea is that the TAs will walk you through this study guide during their review session on Thursday. So the best way to do this is that when I share this study guide, you're going to go and study it, try to understand it, try to find out where your weakest spots are, and then come to this study session trying to learn what the actual answers are and kind of more solidify your understanding of the material. But
Okay,
it really depends. Yeah, it really depends on the person. One thing that you shouldn't worry about, but I actually put it on Canvas for now, is I added some material for the assignment 1. This assignment is due after the exam, so I don't want you to focus on this right now. But I'm just letting you know that I put some stuff there. We will have 2 tutorials next. Is this under syllabus? So we'll have 2 tutorials after the exam on how to read scientific papers. Yeah, so these tutorials are after the first exam. So the exam 1, we have it next week. And then on that week, we have 2 tutorials. These are for the assignment 1, which is due the following week. So I don't want you to focus on that, but I just uploaded those files and then just focus on that. We'll talk about it after the exam, where you have to start thinking about your assignment 1. For now, put your focus on the exam. Okay. Any questions? Okay, I think we left off here, right? All right, you can all still see the screen that I shared, right?
Yep.
Okay. And hopefully you can also hear the sound if I play this video without crashing something.
Hi, Recon. Welcome to our lab.
Can you hear the sound?
Yes, we can.
Okay, great. So let me go back here. So we talked about a few neuroscience techniques, and we talked about a few neuroscience techniques, and one of them, basically neurophysiology, was about recording different areas and finding out what they do. We talked about maybe brain lesion and the assumptions of, oh, if certain area of the brain is lesion and there are some deficiencies in decision making, then well, that brain area must have been involved in decision making. So people, football players that repeatedly have damage to their frontal area because of tackling and headbutting and all that, they end up making poor decisions later in life. Like there are famous cases where they do not make good decisions, and part of the reason is that because their the frontal area is so damaged over time, where the decision making goes wrong, and in a way you're saying, well, this area must have been important for decision making if damage to it is um, so detrimental to decision making. Now, another way to go about this is to actually try to stimulate different areas, right? That's another aspect of, make sure that chat is stuff here. Another thing that, sorry about this. Okay. So another way to find out if a certain brain area is important for something is to stimulate it, right? This is a device called transcranial magnetic stimulation, or TMS. Some of you may have heard of it. What it does is that you could actually hover it over the head, over the scalp, and then zap basically with electrical stimulation certain areas of the brain. And then you could see what kind of movement it causes, right? So, for example, people have actually done this. They can move this thing around. So just in our basic understanding of neuroanatomy, this is the back of the brain, this is the front of the brain, this is the temporal lobe. This is where the parietal area meets the frontal area. And this area people have stimulated that it is the motor cortex. And they have gone as far as actually finding different body parts that move when you stimulate different parts of the system. And I'm going to show you a video of it, of this person actually demonstrating this process. they're stimulating different parts, and you could see different body parts moving.
Laboratory. Today we're going to show you how the transcranial magnetic stimulator works. So first thing, Yoshi's going to put the magnetic coil over top of the area of my motor cortex that's responsible for the muscles of my hand and forearm. Once she's there, she's going to deliver a magnetic pulse. Oh,
So basically, this feels like an involuntary movement because you're stimulating the brain directly, and then it's like a twitch.
and you probably saw my arm twitch there. And we can also see this response over here on the oscilloscope because we've measured it using the EMG electrodes over the muscles of my hand and forearm. Now what we're going to do is move that magnetic coil over the span of my motor cortex from one side all the way over to the next. And what you'll see is muscles slowly recruited from my hand up to my elbow, shoulder, right leg. And after she switches hemispheres, we'll see some of the left leg come in and then down the muscles of my left arm. So here we go. So right hand, little bit of elbow, some shoulder and leg, still right leg, little response in my left leg there, we've switched hemispheres, more leg on the left side, left shoulder, left elbow, and left hand. Thanks for joining us.
So what you saw there is really amazing to me, right? The fact that there are these brain areas where you could actually map that when you stimulate them, certain body part moves. It tells us what? It tells us that there's a specific patch of cortex that is sitting on top. Okay, so let me explain what this cut is, right? So if you cut the brain, like if you take a knife and cut it like this, that's what you get in the left hemisphere, for example, because this is the front. So what we're looking at is the left hemisphere. Behind it is the right hemisphere. And here you could see that you could map out different parts of the body. And as you saw, when the TMS moved down, it went from the shoulder area kind of into the hand area. So she was kind of demonstrating that. Now, you could also map the somatosensory cortex, which is right next to it, which is about sensing. And that you could map out by touching different areas and then seeing the brain activity. So you could go both ways, right? You could have stimulation of external like body parts and see which part of the brain is active, or you could directly stimulate certain brain area and then see which body part moves. And both ways you could find out about functional specialization, right? So how specialized this area is for different body parts or sensing something. And in a way, these sorts of specializations are generally not super strict, right? You can imagine that this brain area is so connected to all the other brain areas that they're all involved in movement, but some specialization exists and partly is due to what's connected to what, right? We talked about this, right? When a certain brain area has a certain function, it's because it's connected in a way that allows it to have that function. Or it's connected in a way that it doesn't have another option. Because if a brain area is the first place that the visual input comes into the brain, it's going to become visually responsive because it's overwhelmed by that sensory input. All right, so TMS is a tool that people use. Yes, Sydney?
Hi, yeah, I just have a question about TMS. So
Sure.
when activating the motor cortex, we see movement. If activating the somatosensory cortex, you said that would activate like tensing, or could you speak to that more?
I was saying in the opposite direction. So, for example, you could do these experiments in cat or rat, for example, where you stroke different parts of their body, or if you could imagine, if we have these body parts, for example, our faces, overrepresented in the somatosensory cortex. Cats and mice, their whiskers are really overrepresented. Each of them has a certain brain area. I was saying if you touch a whisker, for example, or if you touch another, like a human's face, you could see the sensory responses in these brain areas. So I was saying, you're not actually using TMS in this area. You're kind of just stimulating their hand, or not with TMS, just, or as I was saying, if it's in a cat, you're moving their whiskers, and then they're seeing which brain area is most active. So you just
Got it. I
basically,
see. So essentially with the somatosensory cortex, the process would be inverted.
yeah. So
Okay.
in a
Thank
way,
you so much.
the idea is that you're sensing the world, and then adjacent to that, you're acting upon the same thing. So these two things cannot be separate from each other in some ways. As you're sensing the world, that helps you act upon the same kind of, so they're kind of close to each other in essence, brain areas that are processing these activations. Okay, so moving on, we're going to talk about two very common brain imaging techniques. We talked about neurophysiology, we talked about EEG. A lot of the studies that you're going to see done in humans usually uses functional magnetic resonance imaging, or fMRI. And some of the medical field uses PET, or positron emission tomography, and we're going to look at both of them. So because you're going to see these in the papers that you read. So both of these don't really record brain activity. What they record is the hemodynamic response. What is the assumption here? The assumption is that if a brain area is really active in processing stuff, it must be using more blood. So as you can imagine, brain uses a lot of energy, and that energy and oxygen has to be delivered to the blood. And if a certain brain area is really active, it would suck in a lot more fresh oxygenated blood. And if you could actually track that, then we could track brain activity. So it's not a direct recording of activation, but it's an indirect hemodynamic response. This is in contrast with single cell neurophysiology that we saw. Single cell neurophysiology is actually recording action potentials. ERP, which was using EEG or event-related potential, is actually recording a collection, a large population of neurons response, basically. So when we look at PET, the way it works is that because you need more blood in a certain area, the reason the blood goes there is to deliver sugar in a way, right? Sugar is where each cell burns in order to have energy, right? All those ion channels that we saw, all the things, the homeostatic behavior of a cell, all of those things need energy. And if it has a lot of action potential, it requires more energy. So what you do is that you actually inject the patient or the subject with a substance that is radioactive. Not that strongly radioactive, but enough for the radiation to be detected. Then if there is more firing in the brain, these sorts of glucose carrying radioactive units going to be going into the brain. They're going to be picked up by certain brain areas more than other brain areas because they're more active, and then you could record that by looking at the radiation. And the type of images that you get look something like this, where you're actually looking at which brain areas are more active. They're very coarse. They're not really, they give you general ideas. So, when people are in a resting state, you have this pattern. When they're listening to languages or music, they have this pattern. In a way, you get a very general idea of how the brain is responding to different things. So So red generally, I guess, in this images mean that it's more active, blue means it's less active. It has a much better spatial resolution. A spatial resolution in this case means how you could differentiate small patches of the brain from one another, right? You could imagine single cell neurophysiology has the highest spatial resolution because you're listening to a cell or a few cells next to each other. On the other hand, you could imagine when we talked about ERP, event-related potential, you're listening to a huge patch of the cortex. That's what your signal is. This PET has a slightly better resolution. You could actually look at a smaller, say, okay, this is small patch of the cortex has a different response than another small patch of cortex. So what are the pros of this? Well, it has relatively good spatial resolution. The device is relatively easier to use. It's not as expensive in some ways. The downside of this method is that, well, in a way, you're exposing people to radiation. It's a small amount, but I mean, X-rays, and there are a lot of medical procedures that we expose ourselves to a little bit of danger, like harm, for the better good. And this is one of those. But it has relatively poor temporal resolution. So what is temporal resolution? Temporal resolution means that you could say, yes, the activity is from this moment and not from 100 milliseconds later or 500 milliseconds later, right? So there are two types of resolution that I'm talking about, temporal resolution and a spatial resolution. If you can tell me this specific small brain area was active at this moment, then you have really good spatial and temporal resolutions. But if you could tell me, well, this huge patch of neurons were active, but I tell you the exact time they were active, you have a good temporal resolution, but bad spatial resolution. So the concepts are meaning that how accurate is your recording in terms of the amount of neurons that you could actually pinpoint or the exact time point that you could pinpoint. Okay. Moving on, fMRI also is a hemodynamic response space. So, and if you have read papers, you've read about fMRI because they're very common. And what you're really measuring here is also blood intake, but you're measuring the kind of fresh blood. You're saying you want to see how much oxygen is attached to the hemoglobin response, the molecules. So you're just measuring that. The bold signal is, as I was saying, like the other hemodynamic response that are blood related is kind of a slow. So if you show a stimulus and a brain area really likes that stimulus, for example, you show an image of a face, and then you're recording from the face selective area in the temporal lobe, so let's say down here, it takes around 5 seconds to actually, for the activation to be visible in the signal. So that means that you do not have a good temporal resolution. You cannot say, if you have an activity, you can't really say which signal, which image it came from. You could just say on average, this area is responding to these inputs. But when we do experiment in a laboratory, what we do is that you show an image and then you wait 10 seconds, you record the response to know, okay, this image had this response, and then you show the next image. So there's a way to get around it by actually having this inner trial interval that are long in order to get the best signal. And also, it's not super strong. So you get 1 to 2% increase in oxygenation of the blood, and you know this area is very active. That's another point to keep in mind. So what do we do? Well, as I was saying, you could have periods that you don't have any stimulus. You show a stimulus, you have a period that you record that response, and then you move on. Now you could actually subtract the two. Say, okay, brain has activities. that we call resting state activity. That it's just when you're not doing anything, certain areas are active, there are networks. When you show an image, then the activity changes, and then you can subtract that and say, okay, this activity now is related to this image. So what would it look like to have good temporal resolution but poor spatial resolution? Well, as I was saying, EEG has a good temporal resolution, meaning that you're telling me that the activity is the activity of this moment. It's not the activity of 5 seconds ago. No, these neurons are active right now. But you cannot tell me that is it this patch of the cortex or that patch of the cortex. You're giving me activity for a big patch of the cortex. Does that make sense? That's a poor spatial resolution. But temporally, you're telling me that this is the moment. That's why with EEG and ERP, you have, let's say, N200 or N300. These are 200 milliseconds after the stimulus onset or 300 milliseconds after the stimulus onset. You actually know that these two are different signals. Did I answer your question? Okay. All right, so when you look at fMRI signals, what you do is that you actually take an MRI image first, and then you display the fMRI signal on top of that. That way, you could actually see where in the brain the extra activity was. When you look at fMRI images and they look like this, it doesn't mean that only these areas were active. This is the difference between resting state when there is no stimulus and the stimulus condition. That's why you see, and you only show things that are significantly different. That's why you only see a small patches of cortex being active. That doesn't mean that only these areas were active. It's just that the way that we visualize them is just, okay, these areas were significantly more active for this task, or when people looked at faces versus they looked at houses. All right, functional fMRI is spatial localization of it is really good. You could actually, with newer techniques, you could actually pinpoint it to about 1 millimeter cube of the brain. You say, okay, this 1 millimeter cube or 2 millimeter cube has certain activity. You could not get that with PET or with ERP. It's non-invasive, right? You put people, it's a little claustrophobic if you've ever been in the machine, but it's non-invasive and you're not exposing people to much radiation. It has a poor temporal resolution. It means that you cannot really, if you're watching a video, you cannot tell me if I'm looking at fMRI activity, which frame of the video is causing that activity, because the frames are moving fast and the hemodynamic response is kind of slow. The technology is very demanding, it's expensive. In the laboratory, if you want to run an experiment with fMRI, it costs a few $100s an hour. And not all data is good, and then there's a lot of setup, this, that. It's an expensive technology to use, especially for academia. Okay, this is the last slide. Any questions on this? We went through a bunch of neuroscience techniques. Mostly the purpose was that I wanted you to be exposed to them. So as you read papers, you're kind of familiar with them. You kind of know what they're measuring, what the ideas are, because we're going to read these papers, and it's good that you're familiar with them. Yeah, Robin?
Can you explain what what do you mean when you say there's a control stimulus and then you subtract? What's the control? Like, how does that work?
So it depends on your experimental design. Sometimes your control is just the empty screen. So basically there's nothing on the screen, and then you're saying compared to this period, what are the areas that are more significantly active right now?
I was talking about the slide before that one.
That's what I'm talking about.
Okay, got it.
Yeah. Basically, control, you could think of it as simple as trying to account for all the activity that is already there without this stimulus. And saying, okay, this is not interesting. Let me see what is the activity that is only related to my stimulus.
Okay, thank you.
That's the idea, yeah. Okay, I think those are fairly straightforward. Again, you're going to see them in your papers. As you read these papers, you're going to see them. So it's good to know what they are and to be kind of exposed to them.
Sorry, can you explain, how PET has relatively poor temporal resolution?
Temporal resolution, it means that you could actually tell me the activity is from this stimulus that I'm looking at this moment or another moment. If, say, the response is based on how much blood is flowing into a certain area, that's a slow process. So the temporal resolution is low, meaning that if I show you an image and I show you another image and another image, and I'm looking at, let's say, your pet's response, I cannot tell which activity belongs to which image. Like which image caused you to have this activity versus another activity, because the activity is kind of averaged out across time.
Thank you.
Yeah.
So I just have a question about the fMRI. So an fMRI isn't actually looking at anything active. It's just an image of the areas lighting up. So it's not functional in that we're watching brain activity live, correct?
You could watch it live. So MRI is magnetic resonance imaging, and it gives you an image like this. fMRI is functional in a sense that you could actually look at the brain activity. You could actually look at this brain activity based on what I was saying, based on the blood oxygenation level, or the Bolt signal, they call them, right? The Bolt signal. You could actually look at that activity and visualize it. You could also visualize this like a video. You could actually say, okay, what is the oxygen, what is the fMRI signal at this moment? What is it 10 milliseconds later? 10 milliseconds later, you could actually make a video out of it with like 20 frames or 30 frames and then watch it. Is that what you're asking?
Yeah, I think so. I guess to use a more layman's term, is it in real time? I mean, I know what an MRI is. I guess if that's possible, then why does it give poor spatial resolution or poor temporal? Sorry.
It's not real time as it's delayed a few seconds, right? So poor temporal resolution here means if you show a video, the activity is kind of averaged out and you could actually detect it a few seconds later. So 3-4 seconds later, so let's say I'm looking at the face and a house, and you show a face and 100 milliseconds later you show a house. I'm gonna see elevated activity in both the face area and the house area, but I'm not gonna be able to tell you that the house or the face came earlier in the presentation. Does that make sense?
Yes, thank you.
That's what it means to have poor temporal resolution. But if I was recording from single cells in the face area and the house area, then I would be able to tell you which one came before which one, because I'm gonna know that, okay, the face one came earlier because I see elevated activation in the face recorded units, and then later on I see elevated activation in the house recorded units because it's single cell and it's real time. This is kind of delayed and averaged out, so you cannot tell me which one came before which one. That's the idea. Okay, so that's neuroscience techniques. We're gonna talk about neurocomputation. We started late today, so we're not gonna take a break. We're gonna just go all the way out because we have to finish this. We talked about, as I said, there's something, people in this class, as I was saying, we're coming from different backgrounds. But I want you to have this system level understanding or appreciation for the brain, right? The system, and be able to have a mental model of what the brain does. as a computational system, because this computational metaphor is getting stronger every day. So it's important that you have an appreciation for it. And just kind of understand it. It's a complex system, but there are some principles that organizes this system. So it's important for you to have that level of understanding from this organization. This is something from when we talked about this last week, and I think it's important to be internalized, is that all our behaviors, perception and cognition, can be ultimately reduced to a complex pattern of activation within a population of neurons in the brain. Like this is it's a heavy sentence, but the idea is that any state that you have, emotional state, any knowledge that you have, that you recall, any photographic memory that you have, this is all a pattern of activation in the brain, right? There is nothing else. If you look at something and you recognize that face, then it's a pattern of activation in your brain. If you look at them, you don't recognize the face, it's a different pattern of activation. If you look at it, you recognize a house, then it's a different pattern of activation. The idea is that every complex cognition or perception has to ultimately be reduced to a pattern of activation. This is a very reductionist view of it. It's a very scientific view of it. So I think it's important that you internalize it. Okay. So last week, I don't know if this is big enough. Is this big enough? Can you see it well? All right. So last week we talked about a certain type of neuron, and a certain type of networks. This week I'm going to talk about using that motivation. I'm going to talk about networks that we could potentially see in the brain, right? So those were artificial networks that I talked about. They had certain properties, and I wanted you to understand that what the brain does is that it takes one pattern of activation in an area, turns it into another pattern of activation through the weights, and then turns it into another pattern of activation through the weights. The weights are where all our memories, all our knowledge, and all our whatever we've learned is in the weights. And whatever we experience is in the states. So whatever the states of activation is. Now, let's imagine a network like this. And I'm using a slightly different terminal, like visualization here. If a connection is excitatory, I'm showing it with this pigeon leg kind of pattern. If it's inhibitory, I'm showing it with this stop sign and like a roadblock or dead end kind of pattern. So I'm not putting negative and positive on them anymore. For simplicity, I'm using a different notation. Okay, so 1 through 7, these are some neurons. A, B, C are different neurons. And from this figure, we could see that 1 has an excitatory input to A, 2 has an excitatory input to A, 3, 4, 5 all have excitatory input to B, 6 and 7 have excitatory input to neuron C, A and B also have excitatory input to neuron B. So that's a network. It's a slightly more complex than the network we saw earlier, but we're going to build up to more complex networks to understand what happens in the brain. Okay? Just to get some idea, imagine these are photoreceptors, these are interneurons, and these are ganglion cells. So these are kind of inspired by the anatomy of the eye, but we will talk about the anatomy of the eye later. For now, these are just neurons. All right, let's do an exercise. If I have 4, if 4 is activated, has a positive firing rate, right? What happens for B? B would have a firing rate, positive firing rate. If I now activate 3, 4, and 5, what happens to the firing of the B? what happens? You can unmute yourself and answer these questions so we could have it more interactive.
It'll increase.
It will increase, right? Because they are all excitatory to B. So what happens if we continue this? If we activate 2 through 6?
It will increase even more.
It will increase even more because, well, because 2 is exciting A is exciting B, so B must be increasing. And if you go further and activate 1 through 7, it's even going to increase it more. If I activate only 6 and 7, what happens to the firing rate of A?
Nothing?
Nothing, because 6 and 7 are not connected to A, right? There's no connection here, so why would it have any impact? So you could imagine that different cells in the brain have different set of neurons that are connected to them, and through those connections, they have different behavior. Let's see. Alternatively, if you look at a network like this, where you have 1 through 7, and they're connected to a bunch of different cells, and this one in the middle is B. If I activate 4, it's going to activate B. If I increase 4 to 3 through 5, what's going to happen to activity of B?
It would stay the same.
Will it stay the same? Because, well, they're not connected to B. So there is no, what we call is that there is no convergence of activation to B in this network. In this network, there's a convergence of activation to B. So what happens is that as you grow your activation in earlier layers, you would increase the firing rate of B more and more. In this case, you don't do that. Smaller. Okay. So this is called a neuron that has a network that has a high convergence, and this one without convergence. Okay. So I'm going to go back to my video because I think it gives you an idea of what I'm talking. You can all see this screen, right? All right. So I showed a little bit of this video just to give you an idea of what an action potential is. We're going to see some more of it because I think it's very important for getting a sense of what this means. So what do we see so far? So far we see that there is a certain, they're recording from a single cell in the visual cortex. And they see that this single cell is responding to light in this specific area of a space. So imagine a cat sitting, looking at the screen. Okay? So, this is what the receptive field is. For a given cell, you could actually go and then move the light around. I'm going to show you some schematic of it. All right, I'm going to come back to this. So, for example, in the experiment that I showed you, there's a cat. The cat is looking at the screen. There's an electrode in its brain recording from one cell. And this cell, and as you can imagine, the cell is in the back of the brain. That's where the visual cortex is. And you could just move the light around, many locations. That's what the experiment was doing. You move the light around and you could see a single area in a space where the neuron is firing really hard. And that area of the space, you could kind of move it around there. You see, oh, the cat really, this specific cell really likes that area. We call that the receptive field of the cell. So, I'm going to go back to my previous slide. Okay. So you could do that with this experiment, right? Meaning that in this network, you could think of, okay, which area of this space our cell like neuron B really like? Well, if you put it on 1, well, B likes it. If you put it on 4, B also likes it. If you put it on 7, B also likes it. So you could say, well, all of these things are part of B's receptive field. Now, if I take another network A and then put a light here, well, B is not going to have any response based on this light because this light is hitting a different photoreceptor, for example. It's coming from a different place. Imagine these up here are photoreceptors. So just for the sake of, because we're talking about light and stuff like that. You would say that this light is outside of the receptive field of B. So the reason there is a cell that they found that only responds to light from a certain area is because of the way that cell is connected to the photoreceptors in the eye. The photoreceptors in the eye, we're going to talk about that, they are absorbing light, they're transforming it to neural activity, and then that activity is connected to another cells, those activities are connected to another cells, and based on that activity, based on those connectivity, a cell has a certain receptive field, right? So this is something I mentioned last time also. It becomes stronger for B. What is it? I'm not sure. Can you explain the question? So something that I mentioned, if a cell or a brain area is behaving in a certain way, it's because of its connectivity. So if you want to understand the behavior of a neuron, we should look at its connectivity. Okay. All right. So you could imagine neuron B, the neuron that is down here, has a much bigger receptive field than a neuron down here. Because this neuron has a high level of convergence, that a lot of things are being detected by these photoreceptors, and then they're exciting these intermediate neurons, and then they're exciting the downstream neuron. Here, there's a much smaller receptive field where it's hitting the photoreceptors, and then they're just activating one cell. So the different anatomies, different connectivities can give you different receptive field properties. Okay, let's go with any questions so far? I'm going to talk more about receptive fields and show you more examples of what it is. But the idea is that the receptive field of a cell in the visual system is basically the area of the visual world where the cell responds to. If the light is outside that area, this specific cell doesn't care, but obviously there are a lot of other cells that do care. It's just that we're looking from a specific cell and this one doesn't care. That's the idea. Okay.
I have a question.
Sure.
The size of the receptive field depends on the type of cell it is?
Usually it depends on how deep it is in the network. So here, for example, the receptive field of this cell, how does it compare to the receptive field of this cell?
You mean size-wise? It looks the same.
No, the receptive field of this cell. So how many of these things, if you activate this specific cell response?
It's just connected to one.
1 and 2,
Oh.
right? So
Yeah,
okay,
yeah,
so
I see.
the direction is, so this is the input, the middle, and right, imagine this is the output. So this is, let me go back to a better slide for this, so you could see what I mean. Okay, so let's say neuron A. The receptive field of neuron A is only these two. If light hits 1 or 2, it's actually going to impact the response of A, right?
Right.
if you hit light to 4, 5, 3, 4, 5, 6, 7, it doesn't impact the response of A. B, on the other hand, can actually respond if you hit 1, 2, 3 through 7. Each of them get activation, B responds, right?
Mhm.
So the receptive field of A is smaller than the receptive field of B. Like there's a bigger area of the space that B responds to than A. Does that make sense?
Yeah.
It usually has to do with depth. So as I said, the visual cortex is layered. So you have V1, we'll talk about it more, but just since you asked, V1, V2, V4, and then IT, and then there are 3 layers. As you go higher in these layers, the convergence becomes stronger and bigger and bigger, and then the receptive flows become larger and larger. So this really has to do with 2 things. One, how deep they are in the hierarchy of brain areas. 2, how far they are from the fixation point. So human vision or animal vision, right, has a central point and a peripheral point. So I'll show you an example of what that means. So fixate on this, where my mouse is. You see where the mouse is? Fixate here. Don't move your eye and try to read the sentence. How far do you get? How far do you guys get? How many words can you read without moving your eye from the fixation point?
Like 2 or 3.
At most, right? Why is that? Well, because you only have high resolution vision at the fixation point. And that's a result of having a small receptive field. When you have large receptive field, they really average out a lot of activity. So in the periphery, this is something we'll talk more about. But again, I'm just giving you a taste of it. The taste is that when you have really large receptive field, you average out a lot of things. And you cannot really differentiate letters from each other to be able to read. But on the central fixation, you have a really good resolution because you have a smaller receptive field, so they don't interact too much. So there are 2 factors: how far you are from the fixation point, the receptive field, and then how deep you are in the hierarchy. Yes?
Yeah, I have a question. Does it also depend on whether you have, I'm not sure what it's called, but when you have like pluses or minuses in your vision? So like let's say you have like the close-up vision more, then you are able to see less in the receptive field, and then if you have the pluses, then perhaps you're able to see more. Does that depend on it at all?
So receptive field is a property of a single cell. All right, that's not the property of an entire human vision. Does that make sense? We're talking about one cell that they're recording from in that experiment. Another cell, another cell. A cell has a receptive field. The entire vision has certain properties. What you're describing in terms of vision has to do with the anatomy of the eye and the lens and how the lens changes and stuff like that. And you could correct for that by glasses and other stuff. That's the property of the vision itself, not the property of a single cell. I'm talking about single cells, for example, in this network, having different receptive fields. Okay. Yes, Vivian?
Sorry, can you just say that one more time? It depends how far you are from the fixation point and how deep what is?
How deep this neuron is in the hierarchy of the layers.
Okay,
So,
thank you.
are you really close to are you really close to the eye, or are you really deep? For example, as I was saying, in the vision, you have multiple layers of processing, multiple cascade of processing, starting from the eye all the way to, let's say, the frontal area. You go through 7, 8 layers. And as you go higher, the receptive fields become bigger and bigger. Why does it happen? Well, because of this. Because early on, a cell is connected to 2 cells, gets input, and then the cell next to it is connected to those, and then the cell at the next layer is connected to a bunch of Bs and the bunch of, you see what I mean? So as you go, as you stack more layers, it's more likely that your receptive fields becomes larger. Yes, Sophie?
So obviously I can see visually like what the difference is between with and without convergence, but are we saying that like A, B, and C would have convergence, or like would B have like a high convergence? Like, is this representing one cell?
So B has a high convergence, A has a certain level of convergence. So convergence is a more of a continuous thing. It's not just something has convergence, something doesn't, right? As I was saying, if you are in a central, if a cell is responding to input in foveal vision or central vision, it has low convergence because it
Okay.
doesn't average out a lot of input.
Yeah.
As you move away from the central fixation, so if the receptive field is really far from the central fixation, then the convergence becomes higher and higher because the cell gets input from a lot more photoreceptors.
Okay, thank you.
So again, it's a continuous thing. Yeah. So think about receptive field as a property of a single cell, not as a property of vision in general. It's just single cells have receptive fields and you could map them out. And receptive field, because I talked about fixation, it's a complication that you shouldn't worry about right now. Receptive field is always relative to the fixation point. If you move your eye, the cell is not going to respond to the same area. It's going to respond to an area relative to the eye fixation. So if let's say a cell is, let's say my eye is here and the cell is responding to input from this area, if you move the eye, the receptive field has to move with it because the entire visual world is changing on your eye. It's something that comes up later. Okay, so in early visual areas, the receptive fields are kind of circular in shape. So it's a circle in the environment where the cell responds to. And the diameter increases with convergence, right? And I talked about central vision and peripheral vision. When there is high convergence, you have, again, low resolution. What does that mean? It means that if you're averaging out a lot of response, you cannot really tell the difference. The reason I said when you fixate at the word, you cannot read a word that is 3-4 words away in a sentence is because at that location, you really don't have high resolution. You have really big receptive fields. And they average all the visual input, and it makes it hard to actually recognize a word because words are really hard to recognize. The letters are really complex, so you have to really look at it. We'll talk about this more in a few weeks. So I'm going to talk about different types of receptive fields. In the example that I showed, as you could see, all the connections are excitatory. That means that everything that you excite up here, the effect on B is excitatory. So as you make your light bigger, you excite more and more the B. So as you make the light bigger, the B's firing rate gonna go up. So this is basically an entirely excitatory receptive field. But let's look at an alternative. In this case, 1 and 2 have excitatory input to A, 6 and 7 have excitatory input to C, 3, 4, 5 have excitatory input to B, but interestingly, A has inhibitory input to B, and C also has inhibitory input to B. So what does that do? Well, let's start by first, let's say we're probing B. We put our electrode next to B and we want to look at its activity. Well, if I only activate 4 and then expand my light, if I only excite 4 and then expand the light to include 3 and 5. So now we're exciting 3 through 5. Compared to 4, does the activity go up or down? What is it?
Goes up.
Goes up, because 3 and 5 are also excitatory. Now, what if we go a step further? What if we have 2 and 6 also activated? What happens to the activity of B compared to the previous step?
It would go down because the connection's inhibitory.
Yeah, so now 3, 4, 5 are exciting B, but 6 excites C, and C in turn inhibits B. The same goes here. 2 excites A, but A inhibits B. So what does that mean? Again, I'm gonna go with the metaphor we had before. Is A connecting B? Is that a brake or is that a gas in this network?
It's a brake.
It's a brake. When 2 is excited, what does it do to A if it's a pedal?
Yes.
So no, it's an excitatory input to A, but if 2 is excited, A is going to be excited. It means that you're pressing the brake pedal for B. Does that make sense to everyone? This is important. So when 2 is excited, A gets excited. If A is excited, it's going to release a lot of inhibitory neurotransmitter for B. So it's going to bring B down. Yes? Everyone gets this? The same goes over here. When 6 is excited, it excites C because this is excitatory. Now, when C goes crazy, it releases a lot of inhibitory neurotransmitters for B. So together, it would bring B down. Now, if you even further go a step further and actually expand your light a lot, then you're going to even go further down. So let's say you are in this state. What if I remove the light from 4 and just basically give a donut pattern of light? What's going to happen to activity from this state? Does it go up from here or down from here if I remove 4?
Down.
It would go down because I'm removing an excitatory input. Because 4 has excitatory input to B, and removing it is going to even bring B down further. So you can imagine that in a cascade of networks, you have excitatory, inhibitory cascades that can have different impact on each other. If only 6 and 7, so if I give you a network like this that has some excitatory, some inhibitory connection, and I give you 2 patterns of light, you should be able to tell me which one has higher activation. Like you should be able to look at that network and reason that, well, this cell is going to respond more strongly to this pattern of light versus that pattern of light. So a cell like this has an excitatory center and inhibitory surround. That's what you say about a cell like this. The center is excitatory, but the surround is inhibitory. Let's look at some more of this video. I think it's gonna give you an idea of what that is. I'm gonna start over. Initially,
we have a question in the lecture hall.
Yeah.
On the previous slide, do we on the previous slide, do we assume that the numbers are all of equal strength and that the letter strength depends on how many numbers are active?
Yeah, so we assume that they're all equal strength. The weights are all equal in strength. They're either excitatory or inhibitory.
All right, cool, thanks.
Yeah. So initially, they're mapping the receptor though. They're recording from one cell and they're moving the light around, and they found that this area is really the receptive field of this cell. This area is active. Okay, so something interesting happens here. When the light is bigger, you could hear the cell, right? But what happens when the light gets smaller and smaller to a certain size? Here what happens. Can you hear what happens when the light gets smaller? Does the activity go higher or lower?
It seems to go higher.
So, okay, why would that happen? Why is it that from a certain size, when you go smaller, the activity goes higher?
That
Based
like
on-
that like field is like more targeted for the cell where the electrode is.
Yes, but it tells you something about that there's something in the surround, right? It tells you that there's a light that is hitting the excitatory and inhibitory areas, and then when you make it smaller, you only hit the excitatory area. So as I was showing in the example. So what really shows you is that whatever cell they were targeting had an excitatory center, inhibitory surround. That's why when they reduced the light to only this much, they had the highest activity. But when they increase the light a little bit, it's going to hit the inhibitory area and then inhibit the cell down again. So what they found is an excitatory center, inhibitory surround cell. But that the highest activation of a cell like that is only when the light is in the excitatory center. As soon as it's expressed to the inhibitory surround, it's going to bring the cell down again in activity. Okay, so you could actually summarize.
Yeah, we have one more question.
Sure.
You can say it, and I can.
Scream it out, yeah.
I'm trying to connect what you just said to the size of the cell. Like, I'm not understanding why it being so small, it's being activated.
I'm sorry, I don't think I understand your question.
I'm not understanding the relation between the size and the activation.
Size of what?
I think, are you asking about the size of the receptive field or the size of the cell?
The cell in the video. That he had on just now.
That a light getting bigger and smaller.
Yes. Yeah, so I think we're not looking at the size of a cell right now. We're looking at a like a pinpoint of light and how big that light is, so that it is reflecting how much it's on the receptive field. Is that right?
Yeah, so we're looking at light patterns, like so the cat is looking at the screen and then the light patterns are moving and changing shape. And they're finding out which area of the receptive field is exciting this cell by listening to it. You're listening to the firing rate of the cell. Which area of it is inhibiting the cell by listening to the firing rate of the cell. But what we're looking at on the screen is actually the pattern of light. And I was saying, when you make the pattern of light, what they're showing is this, where the pattern of light at this moment, where you're only hitting the excitatory center, the cell is the most active, it's the highest firing rate. When they make the pattern of light bigger, it hits the inhibitory area, so that light goes through, it activates some interneuron that have inhibitory effect on the cell that they're recording from. And that's why the activity comes down again. That's why, I'll show you again. Because when we talk about those networks, they're a little abstract. I'm just showing what it looks like in practice when you're actually listening to a cell. When we're saying they have an excitatory center and inhibitory surround, and what kind of network that we're looking at. This type of network explain the behavior of the cell. So these type of networks explain the behavior of the cell. They're letting you know what is the network in the brain that leads to this behavior from the cell. The size of the cell itself is not important, but what's important is the size of its receptive field, like which part of the world it's responding to. But why would it respond to a certain part of the world? Well, because it's connected to the photoreceptors that get input from that part of the world. Like, I'm just trying to, I think for last couple of weeks, what I was talking about is that in the brain, there is only connectivity and activation. There's nothing else. So everything that you understand in cognitive neuroscience and neuroscience in general has to reduce to that, to connectivity and to activations. And they're not different from one another, basically meaning that one leads to another. The type of connectivity that you have defines what kind of activation you have. So you could understand a lot of diseases in terms of connectivity, and in terms of activation, you could understand cognition in terms of connectivity and activation. That's why we're looking at networks, because that's what the brain is. It's a network. We looked at some abstract networks last time. They were artificial. I said they're the basis for a lot of the tools that we use. This week, we're looking at more biological networks, and we're seeing a neuron of how it responds. Okay, so now you could summarize that excitatory center inhibitory surround in the figure that I'm showing on the right. What you're saying is that if I draw this at some corner of the screen and say this is the receptive field, what I'm saying is that if you put light in this area of the screen, it's going to excite this cell. It's going to increase its firing rate. If you put light in this area, it's going to inhibit this cell. It's going to turn this cell off. It's going to reduce its activity. Those are all the same thing. So you have the excitatory on center and inhibitory off surround. For one cell, it could be in this area with this property. For another cell, it could be somewhere else with different property. So it's important to understand we're talking about single cell receptive fields. And this cell down here would have that response, right? It's a slightly different network that I'm showing, but you shouldn't be surprised by it because, well, if there's activation here, it excites this, then it excites that, and this one inhibits this. Same here. Here it's exciting this, and then it's exciting that. So the center is excitatory and the surround is inhibitory. You should be comfortable with just looking at a network and knowing, okay, this is what's happening in this network. But this is a different way of visualizing the same network. in the terms of the effect that the light has on the neural response of this cell. So you're kind of ignoring all these other cells and only describing the behavior of this cell using this receptive field visualization. That's the idea. It's just a visualization of what you see on the left. So what would be an optimal stimulus for this cell? Well, something like this, where you have light only in the excitatory area and you have nothing in the inhibitory area. That's a light that really drives the cell crazy. Like it's gonna fire really hard.
What would be a cell, what would be a stimulus? I guess it's just a bigger circle would calm it down a little.
Calm it down. If it was a ring, like a donut or like a bagel, then that would really inhibit this cell. It's kind of basically quiet this cell.
But would it, I mean, how do you see a bagel then? There's some other center surround organization that's the exact opposite of this?
Yeah. So there are other cells. You mean other cells that have different receptive field?
Yeah. Or
There are,
that gets stimulated in the opposite way, such that in the middle it's negative, on the outside it's positive.
Yeah, so that you have off surround, off center, on surround cells. Yes.
Right.
Yeah, you have all sorts of combination of cells, different receptive field sizes, different receptive field locations. You have everything in the brain. Like in the visual cortex, we'll talk about this, the fact that you're detecting these things all over the visual field with different cells, that they have different properties, that you could actually detect all sorts of things. Yeah, that's how you detect the ring. And then kind of, we'll talk about this hierarchy and how you build up more complex until, let's say, you arrive at a face. But you have to start with detecting the small circles. And then from that, maybe you could detect edges. Then from there, you could detect corners, and then shape, and then objects, and then face. Right? It's a hierarchy that you build in order, as the receptive field gets larger, it also becomes more complex. But for now, we're just talking about very simple behavioral from the cell. We're talking about just detecting a single, basically circle. That's it. That's all we're worried about. Like a circle of light can actually be detected by a cell, and it responds to the rest of the brain saying, okay, I detected a circle of light in this area of the world. Okay, so the receptive field, as I said, is the region of the visual field that one cell sees. This is one cell. There are millions of the cells in the visual cortex, and they all are looking at different things. They're roughly circular early on, and then later on they become more complex. The size of the receptive field depends on a few factors. I mentioned a couple of them, and we're going to get deeper into that later on. And it has the center surround antagonistic organization, where it's a product of this lateral connection. We're not going to get into that. And as you mentioned, as the question was, there are cells that are inhibitory center and excitatory surround, and you visualize it differently. Let's imagine, well, what would you change in this network to get the other cell?
You would just switch the ones that are surround. You would make the surround in this part excitatory and the center inhibitory.
So how many changes would you have to make here? How many connections would have to change?
Well, the two at the bottom would have to become in excitatory, and then the one in the center would be inhibitory. So 3.
Exactly. So if I make these two excitatory on each side, and then this one inhibitory, then I'm going to get the opposite receptive. Because the center becomes inhibitory, the surround becomes excitatory. Simple as that.
And that has a different name, or
So yeah, we call those basically off-center, on surround. Yeah. Because you kind of, the center kind of turns off the cell if there's light in it, and the surround kind of turns it on. If you have more time toward the end, I'll show you more of the video. They also detect those cells. It's just that this research kind of, the way to think about the brain this way, a lot of it started from that line of work that I'm showing the video from. That's why I think it's a classic and it's important to know about, because a lot of these way of thinking about networks, it's been very influential. And also, this has been the basis for a lot of accomplishment, at least what we see more fruitful recently in machine learning and artificial neural networks. This idea of you have a network, and the network has to build up complexity across layers. You can't just have complexity right away. No, you have to start this detecting the small things, then from there detecting more complex things, and then build up through a network. Because in reality, these three cells that I'm showing in the middle, they're also simple detectors. They're only detecting a small patches of light, because they're all like, all the connections from these light detectors to them is excitatory. So they're detecting the small patterns of light, but the way they're connected to this cell creates this interesting circle detector. So you could see this complexity building up here even. Yeah, so inhibitory center, excitatory surround. That's the other name for it. All right. So you could imagine the type of cells that we saw are very early on, very close to the eye. And again, close in this sense means in the cascade of neural activity. When the light hits the eye, it takes about 100 milliseconds for you to recognize it as a face, for example. In this 100 millisecond, it goes through 10 maybe cascade of activation. So it goes from the eye to, let's say, the LGN goes to V1, and then you go to V2, V4, posterior, middle, anterior IT, and then you go to frontal area. So it goes through this cascade of brain areas, and every time it becomes, the patterns becomes more and more complex. Like when you go higher up, you don't respond to small things, but you respond to really complex objects. So you could imagine that we could have more complex patterns of receptive fields. Rather than a circular center surround, you could have elongated center surround, where the center area is excitatory and the surround area is inhibitory. It's also center surround antagonistic, and you could imagine the opposite of that would also exist. And these cells are in V1. They're like a couple of stages further from the eye. As I said, you could have on-center, off-surround elongated receptive fields, or off-center, on-surround elongated.
Sorry, would this just be like depth perception? This elongated?
No, this would be edge detection. Depth perception involves the two eyes that we'll talk about a little later. Depth perception is about when, so one thing that the visual system does is that you take actually the visual input from the same brain area, from the same visual area, from the two eyes, and you just take them next to each other in one hemisphere. So for example, if I'm looking at something in my right visual field, I actually have the visual input that comes to the left eye and the right eye go to the left brain because it's on the right visual field, and they're next to each other. The disparity between them is used for depth perception because the input from the two eyes are slightly different, and that disparity is important for depth perception. These elongated receptive fields are for edge detection. So you could detect an edge, basically an orientation in space. I'll show a video, you'll see what these cells are like. call these simple cells. So again, similarly to you saw, you have a pattern of light, and then you move it around. And in this time, it's not just a circle, but elongated pattern of light, like a rod. So first, they're mapping out the excitatory area. When they show light in this orientation, in this part of the visual scene, it gets really exciting. But when they show it in this area, you could see that the cell goes quiet. You see that when the light comes in this area, the cell practically goes quiet. That means that that part of the world is inhibitory for this cell. Okay, so you can actually see these cells in action like that, where you could actually take a pattern of light, you orient it, and you move it around, and you see, well, now the receptive field is not a simple circle surround that you say, okay, it's this part of the world with this size. There are more properties. It has a certain orientation. Yes, Robin?
The only question I have is, you just said that area of the world is inhibitory. What if it's just outside the receptive field entirely?
Then it doesn't have any impact on the response of the cell.
So what's the difference between inhibitory and just not even in the receptive field?
Inhibitory means that it silences the cell. So,
Right, but like on that thing, would you hear noises? I mean, you'd hear fewer noises because the base rate is lower.
it's lower than the baseline activity.
So if it was outside the receptive field, it would be slightly higher because it would just be base rate.
Yeah.
Got it. Okay.
And then if it's in the inhibitory area, it inhibits beyond that. So for example, in the networks that we had, if the light was only in the air, so if there was no light, you would have a baseline activity for B. But if you only put light in 1, 2, and 6, and 7, then that activity would become lower than the baseline because you're only inhibiting the cell. So that's the idea.
Got it.
Okay, so you could actually now search for a given cell, you could search for these orientations that the cell responds to. And basically have a stimulus, move the stimulus around, and then you could actually have and see, well, wow, this cell really likes this pattern of light. Now, I've been using different terms like, oh, this pattern of light or this input really excites the cell, or the cell really detects this, or it's tuned for this, or it really likes, but all of these things mean the same thing. It means that this pattern, when it's in the input, if it's in the receptive field, this cell is going to get really high firing rate. That's what it means. Now, you can imagine for any cell, they're combining excitatory and inhibitory stuff. So in this case, the most excitatory input would be a pattern of light only here. So what if you rotated your pattern of light a little bit? What would happen to the activity compared to the case that the pattern of light is only in this area? What would happen?
It would inhibit.
It would go down a little bit from that maximum, right?
Because
you're
part
now
of it's crossing it.
Yeah, because part of the light is in the inhibitory area, so eventually brings the cell activity down. So we're not moving away from that initial idea that we had, that every cell gets a lot of inhibitory and excitatory inputs, and it combines them, and that combination defines its activity. So when you have this response, you're really active. When you have this response, you're not active at all. When you're something in between, you're somewhere in the middle. Because at this point, you have some excitatory stuff, some inhibitory stuff, but maybe a little bit more excitatory, so you're still a little active. In this case, you have a lot more inhibitory than excitatory. Like when you, for example, in this receptive field, if your light is horizontal, you get a lot of inhibitory versus excitatory input, because the other excitatory areas are not activated by the light. Okay, so the simple cells have these elongated receptive fields rather than circular, and they respond to stimuli having a particular spatial orientation at this point, right? They're not just detecting light in certain areas of the world, they care about the orientation now. What does orientation get you? Well, orientation gets you edges, because the world is made of edges, right? The table that I'm sitting in front of has a bunch of edges. For me to actually detect this table, I have to detect the edges, and that's important. And then edges become corners, and corners become shape. So you build up from there. So it's important to understand this hierarchy of representation. Simple receptive fields are constructed by selectively combining the circular receptive fields. All right, let's imagine a network where this is possible. And this is the last thing I'll cover for today. So if you have a bunch of a bunch of antagonistic center surround receptive fields. So these are different neurons, and they have these receptive fields. If you combine the response of a bunch of them that are like this, they're going to give you a receptive field like this, right? I want you all to understand this. Why? Because, well, if there's light here, it's going to excite this cell. If there's light anywhere in this area, it's generally going to excite this cell. And if there's light around it, it's going to inhibit this cell. So you can imagine how combining responses from these center surround antagonistic cells can actually give you an oriented simple cell based on this connectivity. So based on this hierarchy. If it was a bunch of other cells and they were connected to another cell, as you can imagine, there are all sorts of connection in the brain. So there are all sorts of lower level cells that are center surround antagonistic, and those cells' response are being combined to get the elongated receptive fields. If it was a bunch of other cells in this direction, you would get a different simple cell. So every simple cell is practically a combination of a bunch of antagonistic center surround cells. And that's how things work in the brain. Every cell is combining responses from other cells, and based on the way that it's connected, it has a certain problem. Does this make sense to everyone? Any questions about this?
Yeah, so is that two different ways of creating an orientation, or you're just saying that the circles lead to the top? The top isn't a separate
this is
option.
the only way. This is not a separate option. You cannot arrive here immediately. You have to go through this process. You have to first detect a bunch of light next to each other, and then somebody else, another neuron is connected to this bunch, can actually detect an edge. You have to go from this level to that level. There's no direct path to just detecting an edge. You have to actually detect every single piece of the edge and then connect them. That's what that means. So this slide just says that it's basically detects oriented edges and bars. But let's spend some time here, however much time I have left. 1 minute of time that we have left. So the idea is that you are building hierarchies. And through these hierarchies, the network can detect more and more complex things. And we build up on this hierarchy as we go through this course until we arrive at more complex stuff, obviously, like recognizing faces and what attention is and all those sorts of things. But think about how this connectivity in this simple case of having edges. So when I say the receptive fields like these, each of these is basically a neuron. I'm just representing them in this way. This is one neuron that responds to this way in the wall. There's light here, it's excited. There's light outside, is inhibited. Same here, same here. And then when a cell that is and imagine these actually are tiling this space. Somebody has to move in the main room so it turns back, or the keypad. Yeah. So imagine that these simple cells, these antagonistic center surround cells are tiling this space. They're everywhere in this space. And a bunch of them are connected to a simple cell. A bunch of other ones are connected to another simple cell. A bunch of other ones are connected to another simple cell. And the way they're connected and which one of them are connected defines what the simple cell properties would be, like which orientation it actually responds to. Any questions about this? So I hope today, what would it? No question in the chat. I hope today you got an idea of why we talked about networks. Why was it important to think about networks? And why is it important for you to get an intuition of excitatory inputs, excitatory connections, inhibitory connections, and how these combination of excitatory and inhibitory connections do lead to different behaviors of the cells. Okay? Does that sounds good to everyone? So next week you have the exam. Just study your study guide, show up to the TA session. And if you cannot show up, it's going to be recorded. You can watch the recording. I'm sure they're going to give you a lot of insights. As I said, they all had the course. And they TA'd for the course too. One of them has TA'd a few times. So they're very knowledgeable and they're the nicest people that I've met. So you should go there, you should ask for their help, and I'm sure they can help you get a better sense of these concepts. It's completely normal if some of these concepts come to you not in the easiest way, because this is a different way of system thinking, of computation and engineering and those sorts of things. So it's okay. Just do your best, and I'm sure you're gonna do well. All right. Okay. Stop recording.
Thank you so much, Professor.
Sure, thank you.